Now we will be talking about elongation process in details. Okay. Now, when you are talking about elongation process, you must understand that suppose this is your double-stranded DNA. Okay. Suppose this is your 5' end, this is your 3' end, and this is your 5' end, and this is your 3' end. Okay. Clear? So you must understand that this 3' region will be actually the point from where RNA transcription will start. This 3' region is actually the point from where RNA transcription will start. Clear? Now listen with all your attention. So if this 3' portion is the place from where your RNA transcription will start, then you must understand this very basic thing that the RNA which will be produced, suppose this is the RNA, this red one is the RNA, R for red, R for RNA. Okay. Now listen, this RNA will be having a polarity of 5' to 3', right? Now listen, the RNA polymerase actually recognizes one of these, strand. The RNA polymerase, recognizes the strand which is also known by the name of template strand, right? Which is also called template strand. Clear? Which is also called template strand, and this is non-template strand. Now, the strand opposite to the template strand, that means this strand, is called coding strand. Okay, is called coding strand. Is called coding strand. Okay, so this is what coding strand, this is template strand, and this is what coding or non-template strand, right? Now listen with all your attention. A very important point if you understand here, that is RNA polymerase reads only one of the strand, but takes 2 strands at a time, right? Takes 2 strands at a time. Now listen with all your attention. Suppose this is our RNA polymerase diagram. This is what NTP channel from where ribonucleotides enter. Clear? Now listen, this is NTP channel and this is what RNA exit channel. This is what DNA exit channel. This is DNA entrance channel, right? And this is the channel, or this is the place actually where unzipping of DNA occurs. Now listen, suppose this red strand is the template strand, okay, and this blue strand is the non-template strand. This red strand is the template strand and this blue strand is the non-template strand. Clear? Now a very important point you need to understand here, is this, that suppose this red one, this is what, suppose this is your 3' end, this is suppose your 5' end. Okay. Now listen, that RNA polymerase has a special function, okay, that is they do not require any primer for their initiation of transcription. Now listen, suppose this this RNA polymerase has started what? Has started transcription, okay. So that means it will be now listen, in a DNA, I just want to make you people understand one basic thing, that is in a DNA both the strands are facing each other, right? 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 Now important point here we need to understand is this, that when you unzip this DNA molecule, the first thing you need to understand is this: when you unzip this DNA molecule, what this RNA polymerase is doing in details, you need to understand here. this RNA polymerase is doing an extremely, important thing. That is, what? Suppose you unzip this DNA. Suppose with the help of heat you unzip this DNA. Suppose. Now listen with all your attention. What is the difference between this unzipping and unzipping by this RNA polymerase? Now listen with all your attention and look at the diagram. If you unzip this, RNA DNA polymerase, sorry, RNA DNA hybrid, sorry, only DNA, then what is the difference? Now listen with all your attention. Here again, what are these? These are your nucleotides, right? These are what? Your nucleotides, right? This is what? Nucleotides. This is what? Nucleotides. Now, a very important point you need to understand here is this. A very important point here you need to understand is this, that the red nucleotides, or the nucleotides which are attached with the red, strand, the red nucleotides actually will not only unzip themselves with the help of RNA polymerase. What they will do is, now listen. Suppose this is your, you thought this will be the, unzipping pattern, right? That means this is what? These are your unzipped nucleotides, right? But what RNA polymerase in this case does is a really exceptional thing. You know what it does? It will actually flip the nitrogen bases out. It will flip the nitrogen bases out. Yes, that it will do. Okay, it will flip the nitrogen bases out. Clear? It will flip the nitrogen bases out. Clear? It will flip the nitrogen bases out. Okay, it will flip the nitrogen bases out. So in a normal denaturation, what you are seeing, the nitrogen bases are still, pointing towards each other, right? But in a RNA polymerase, related denaturation, what you will see that, or unzipping, not denaturation, or unzipping, what you will see, these, unzipped nucleotides are flipped outwards. This is one of the basic functions of RNA polymerase. Now, what it will do, now listen with all your attention. When these things are flipped, then now listen. From here, RNTPs will start entering. RNTPs will now start entering, and RNTPs will now interact with these flipped nitrogen bases. RNTPs will now start interacting with these flipped nitrogen bases. Clear? Now listen with all your attention. When RNTPs are interacting with these nitrogen bases, flipped nitrogen bases, then RNTPs will be, joined by releasing pyrophosphates by RNA polymerase. So these are the two, main functions of RNA polymerase during RNA transcription. That is, it will flip the nitrogen base, okay, in elongation phase. Obviously, most important functions of RNA polymerase in elongation phase is this, that it will flip the nitrogen base first. Second, it will, can you see that? Okay, now listen, it will flip the nitrogen base first, number one. Number two, it will now allow the RNTPs to enter into the RNA polymerase, this domain. Okay, now what happens is these RNTPs will now join with the flipped nitrogen bases. Okay, suppose there is, A, G, C, T, A. So instead of A, here will be U, C, G, A, U. U, C, G, A, U. Okay, U, C, G, A, U. Okay, now listen with all your attention. A very important point here we need to understand here is this, that whenever you are doing these things, whenever RNA polymerase is doing this, not you are doing, whenever RNA polymerase is doing these things, now a very important point you need to understand here is this. Suppose this is, suppose this is what? This is what? RNTP, okay? Suppose this is RNTP, okay? Now listen, whatever be the base, okay? Suppose this is RNTP, okay? Ribo, let me write like this. Suppose this is an RNTP. Now listen. Now important point here we need to understand is this, that to form an RNA, this phosphate will now have to interact with these, another RNTP, then again another RNTP, then again another RNTP, right? Now a very, very important part is this. Now this is what? If this, polymerization takes place with the help of RNTPs, then it will be RNA, right? Now listen, suppose these are RNTPs, okay? Now what I am saying is this, that this bonding is actually making RNA, polymer, right? These bondings are actually, helping RNA to form an RNA polymer, or RNTPs to form an RNA polymer. This is the function of RNA polymerase to form these bonds. Clear? When they unzip this DNA and flip the DNA bases out, okay? So this is the basic thing you need to understand. They are flipping the bases out so that, corresponding RNTPs can actually interact with these, flipped bases. But they are also doing one thing, that they are actually joining these bases, okay? They are joining these bases. These are the bases, okay? These are the bases. These are the bases. That is, these are these things, okay? So this is also done by RNA polymerase, okay? Clear? No problem up to this, okay? Now a very important point you need to understand is this, that during elongation, that DNA polymerase have proofreading function. But do you know that RNA polymerase also have proofreading function? Yes, they can proofread it. They can proofread whatever mistakes they are, making. But their proofreading is actually not very much, good. Therefore, it is said that RNA polymerase, performs more mistakes with respect to, DNA polymerase. That means the proofreading of RNA polymerase is not good as that of DNA polymerase. Now, we will understand in details what are the types of proofreading RNA polymerase is using, okay? Now listen, there are actually two models of proofreading mechanism of RNA polymerase, okay? There are two models of proofreading of RNA polymerase, okay? Of proofreading in RNA polymerase. Okay, there are two, models. Now, what are those models? Now, listen. The first one is called kinetic proofreading. Okay. And the second one is called nucleotypic proofreading. First one is called kinetic proofreading, another one is called nucleotypic proofreading. Now listen with all your attention. A very important point you need to understand here is this, that what is kinetic proofreading and what is nucleotypic proofreading. Now listen, there are many, explanation about that, but the easiest explanation is this, that suppose I am an RNA polymerase, okay? Now if suppose, this is suppose, another, this is, imaginary suppose DNA template, okay? Now suppose if I am inserting RNA polymerases, okay? Sorry, if I am inserting RNA prime nucleotides, okay, RNTPs. Now suppose this is the position 1, this is position 2, this is position 3. Suppose I have inserted a correct RNTP here, I have inserted wrongly a wrong, RNTP here, and again I am moving. But suddenly I realize that, oh, here is G, but I have inserted U, right? So what I can do is, I can actually, cleave that particular RNTP and then I can insert another, correct RNTP. Clear? Now, a very important point you need to understand is this. This is, kinetic proofreading. That means, suppose this is, your DNA, and suppose this is, RNA, bubble. Clear? Or transcription bubble. This is called transcription bubble. Okay, and there are flipped bases here, right? Suppose this is A, G, T, C, C, G. So the RNA will be having, C, G, G, A, C, U, right? But suppose by mistake RNA polymerase inserts here U. So this is wrong. Now listen with all your attention. What will happen is this. Now listen. What will happen is this. Now you need to understand that suppose RNA polymerase. Okay, sorry. So now we will be discussing kinetic proofreading. Okay. Now suppose RNA polymerase is now starting to incorporate nucleotide base here. Okay. Now suppose it has incorporated here what? C. Okay. Now what will be this? G, right? What will be this? G. Here what will be this? Opposite A, right? Now listen. Suppose by mistake instead of A, RNA polymerase incorporates U here. Or C here. So this is a mistake, right? Now listen, what will RNA polymerase do? RNA polymerase will actually cut this bond and will remove this C and insert, and will insert A, right? And will insert A. Clear? And will insert A. Clear? Now, a very important point. What is nucleotypic proofreading? Now, important point you need to understand here is this. What is nucleotypic proofreading? There is no basic difference between nucleotypic proofreading and kinetic proofreading. But the most important difference, what is that? The most important difference between nucleotypic proofreading and kinetic proofreading is that in nucleotypic proofreading, the mistake is realized with RNA polymerase in that moment only. But in nucleotypic proofreading, what happens is this: RNA polymerase has done that mistake, okay, and is continuously adding correct RNTPs to other DNA templates. But suddenly it realizes that no, there is a mistake. Suppose, let me explain. Suppose this is clear? So it will be flipped out. Now listen with all your attention. Suppose this is what? A, C, G, G, C, A. Suppose. Okay, let it be T. So here it is T, G, C, C, G, A, right? Now important point you need to understand here is this, that suppose we have got this sequence here. Now what is nucleotypic proofreading? Now suppose again this is an RNTP, what? This is uracil, this is another RNTP, right? What is this? C, this is another RNTP, what is this? G, this is another RNTP, what is this? G. Now instead of G, suppose RNA polymerase has entered U, suppose, and it has gone to this section, okay? What is this? C, U, right? Now listen. So this is an error. So this is an error, right? So this is an error. Now important point here we need to understand is this, that whenever this type of polymerization occurs, then there is what in between these bases, what is there? Hydrogen bonds. Now listen with all your attention. Now suppose RNA polymerase, actually suppose here is your RNA polymerase. So this is DNA entrance site. Clear? This is your DNA entrance site, and this is what? This is what? This is what? RNTP, entrance channel. This is RNA exit channel. So what I will have to do here is this. I will have to make a correction here. What is the correction? Simple. The correction is really simple. You know what is that? But it should be done for your betterment. Now listen. The correction is very simple. That is, instead of, what will be this? Instead of G, there will be C. And here will be U, right? So it is now going in out of the RNA polymerase in exit channel. Clear? Now important point here we need to understand is this, that suppose this is the picture of your RNA polymerization, but important point here, what is there? Here there is what? Here there is a mistake. Now listen, RNA polymerase, what will this do? Now listen, many people think that RNA polymerase will go back. No, listen, the DNA is coming into the RNA polymerase, it will not go back or something like that. Now listen, suppose what will, now listen, suppose it has gone, suppose the mistake is in this part, suppose the mistake is in this part, okay, then it may stop the polymerization reaction and it will do something extraordinary. Okay, there are many protein like TRCF or something like that which will actually interact. We'll talk about that later, but now listen with all your attention. Suppose after some incorporation of nucleotides, it has realized that there is a problem. What it will do, it will dismantle all the hydrogen bonds here. It will actually dismantle all the hydrogen bonds here. Okay, it will dismantle all the hydrogen bonds here, and it will now cleave this portion, this bond, and it will get away, it will replace this, only this portion of RNA with G. That's it. So this is nucleotyping. That means what? When a very good number of nucleotide has been incorporated into RNA, then nucleotype proofreading takes place. But when the mistake is, the mistake has just happened, then kinetic proofreading takes place. Clear? Okay. So now we know what is proofreading, okay, of RNA polymerase. But a very important point you need to understand here is this, that we have talked about, RNA polymerase proofreading. But if there is a mutation or there is a problem within the DNA itself, okay, if there is a mutation or problem in the DNA itself, then what will happen? Now listen, if there is a problem in the DNA itself, what will happen is this RNA polymerase will be stopped by a protein called TRCF. Clear? TRCF. Okay, TRCF. Now listen, what will happen? When this TRCF interacts with this polymerase, it will actually push RNA polymerase. It will actually push RNA polymerase the same way raw proteins work. Clear? This TRCF protein will push RNA polymerase. Now listen, RNA polymerase has two choice after that, after this pushing, initial pushing by TRCF. Okay, it will now what? It will now push RNA polymerase. Clear? Now after this pushing, this RNA polymerase has two choices. Number one, it can actually go on with the transcription, and another choice is it can now fall off. Clear? This is how, if there is a problem in the DNA or mutation in the DNA, there is a large amount of mutation in the DNA, then RNA transcription may halt. Clear? This is how RNA transcription halt if there is a mutation in the DNA. Clear? Now, how this TRCF protein is activated. This is actually a part of DNA correction mechanism or RNA correction mechanism. We'll talk about repair systems later. Clear? Okay. Now we need to understand another thing. Now we will be talking about termination of transcription in prokaryotes. Now we will be talking about termination in prokaryotes. So this is termination in prokaryotes. Now listen, termination in prokaryotes actually have two well-defined mechanism. Number one, row dependent number two, row independent. Clear? Row dependent and row independent. Clear? So in prokaryotes, transcription occurs with the help of row proteins, or it may be row independent as well. That means no row protein is required. Clear? So there are row dependent mechanism and row independent mechanisms. Clear? So there is row dependent mechanism and row independent mechanisms. Clear? Now listen with all your attention. A very important point here we need to understand is this, that in case of termination, we'll first talk about row dependent mechanism. Okay, then we'll talk about row independent mechanism. Clear? Okay, so let's start with row dependent mechanism. Okay, so let's start with row dependent mechanism. Now listen, in case of row dependent mechanism, you know that row protein will play a very important role, right? Now listen with all your attention. Again, we'll have to draw our polymerase. Now listen, we are always drawing like this, but in this drawing or in this diagram, alpha helix, alpha subunits are absent. Okay, so this is actually beta pleated, beta sheet in details. Sorry, not beta sheet. Beta and beta prime pins are detailed here. So this is suppose your clear? Now listen with all your attention possible. Now listen with all your attention possible. So we will be talking about row dependent termination, right? Now listen with all your attention. Suppose this is your so this is 3' of DNA, this is 5' of DNA. So therefore, what is this? This is 5' of RNA and this is 3' of RNA, right? Now listen, in the 5' side, now listen with all your attention. In the 5' side, there is an important point you need to understand. If you want to end transcription with the help of row protein, you need to have this site. Now listen, there is a special site in 5' end of RNA, which is coming out of the RNA polymerase. Okay, this site is called RUT site. This site is called RUT site. This site is called RUT site or row utilization site. Okay, this site is called RUT site. RUT site, okay, or row utilization site. Clear? Or row utilization site or RUS site. Okay. Now listen, Actually what happens is this rho protein, this rho protein is actually a hexamer, okay, and it looks like a donut, okay, 1 2 3 4 5 6, okay, clear. So this is an hexameric protein. Now listen with all your attention. This, what is this rho protein? What is the chemical nature of this rho protein? Rho protein is nothing but an helicase, okay, it's an helicase. Okay. Rho protein, this is rho protein, and this is nothing but a helicase, clear. Now what happens is this rho protein actually binds with this right side, sorry, not right side. This rho protein 1 2 3 4 5, this rho protein now binds with the right side, okay. Now after binding with the right side, what happens is this helicase utilizes ATP, okay. This rho protein, these are the characteristics, okay. They utilizes ATP, okay. They utilizes ATP. Now what this particular rho protein is doing is, this rho protein is actually utilizing ATP, to move towards the this RNA. Now what happens is this, like TRCF protein, I have already told you, like TRCF protein, this rho protein actually, what it does is, this rho protein actually gives a blow to the RNA polymerase. It actually literally gives a blow to the RNA polymerase, clear. It gives a blow to the RNA polymerase, and therefore RNA DNA, this transition, or RNA DNA, this bonding actually breaks off, and the RNA which has been transcribed falls off, clear. Now so when this rho helicase actually now interacts with RNA polymerase, it gives a blow. Now another mechanism is this, that with the help of ATP, with the help of energy derived from ATP, rho actually moves into this site where there is RNA DNA hybrid. This is the site where there is RNA DNA hybrid. Now this is what? This is RNA DNA hybrid, right? This is RNA DNA hybrid, right? Now a very important point here we need to understand is this, that this RNA DNA hybrid actually gets dismantled or is disrupted when rho helicase actually arrives here, and therefore this RNA DNA hybrid breaks off and RNA comes out of this RNA exit channel, and termination of transcription takes place, clear. Okay, now listen with all your attention. What about this Rus site? Now Rut site, sorry, why I am telling Rus site? Now this Rut site is rich in CA, that is cytosine and adenine, clear, is rich in CA, clear. Okay, so this is rho dependent mechanism. When you are talking about rho independent termination, you need to understand that for rho independent termination there are two prerequisites. Okay, now listen, those prerequisites are already present in the DNA structure itself, clear. Now listen, suppose, so this is rho independent mechanism, clear. Now listen with all your attention. To understand row independent mechanism of RNA transcription, you must understand this very basic thing that there are two prerequisites. Okay, I have already said that. Now listen. These prerequisites are present on the template strand, but they are also present in the non-template strand. Why? Listen. So, what are the prerequisites? What are the prerequisites of row independent mechanism? The prerequisites of row independent mechanism. Now listen with all your attention. The prerequisite number one, is very, very important. That there should be alternative base pairing having a gap. Okay, there must be alternative base pairing. Alternative base pairing should be there. Okay, alternative base pairing. Clear? Alternative base pairing. And the second one is the presence of repetition of A rich site in the upstream of DNA. The second point is repetition of A rich sites upstream to the alternative base pairing. Now listen, what is this? Suppose there is TC triple G CG, and then it is followed by N. N means any nucleotide. Okay, N means any nucleotide, and G. Thereafter CG triple CG. Now listen with all your attention. A very important point to understand. And in the upstream, okay, in the downstream, there are A. Okay. Now listen with all your attention. Whenever RNA polymerization will take place, now listen with all your attention. Whenever RNA polymerization will take place, now what will happen is, whenever RNA polymerization takes place, what will happen is, there is what? C, G, 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 C, G, N, right? And then again it is C, G, 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 C, A. Okay. Now listen with all your attention. Now if I can draw the transcript here, you will understand that, suppose this is your transcript. Okay, so we have got C, G, 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 G. There are 3 G's, and C and G. Then after there are nucleotides again. Then there is C, G, 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 C. Now listen with all your attention. Can you find the peculiarity here? Now listen. What is the peculiarity? This C can now interact with this G. This G can now interact with this C. Clear? And sorry, I have put here okay, I have put here G's, so this will be C, opposite to G there will be C, opposite to G there will be C, opposite to C there will be G. Okay, so there will be 3 C's here, sorry, clear? Now listen, so can you find the peculiarity here? Now listen, what will happen is this, now this whole thing will be converted into a thing like this loop like structure. You know why this loop is occurring? You need to understand this. You know why this loop is occurring? Now listen, this loop is occurring because, now listen, suppose this is your mRNA. Okay, now listen, suppose this is your mRNA. Now there is what? G, then C, right? Then G, 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 C, right? And opposite to C there will be G, C, 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 G, C, right? You will get these things here and here. That means these two are complementary to each other. Therefore, they will have what? They will have hydrogen bonds. Now, important point you need to understand here is this. This loop consists of all n nucleotides. Okay, that means any nucleotides. Clear? But when this stem loop-like structure, or hairpin loop-like structure takes place, now important point you need to understand is this, that when this thing actually comes out, and forms a hairpin-like structure, and forms hairpin-like structure, then you must understand this very basic thing. That means this whole thing, that means what is happening here is this whole these A things have actually now come here, right? These A's have come here, right? Now when these A's are coming here, that means you have a nasty mind, you are laughing now. That means you, right? Now listen, due to this weight, now listen, this is very important, okay? This is very important for row independent, termination. Why? This continuous, repetition of A nucleotides or adenine nucleotides are very important, because these adenine, when it interacts with you, they will have what? Double bond, and double bond is weaker than triple bond. Therefore, when this stem loop or hairpin loop like structure will occur, then due to the weightage of these loops, these hydrogen bonds, these two hydrogen bonds cannot hold this heavy thing, and therefore it will fall off. Therefore, this whole thing, this hydrogen bond will break off, and this will all fall off from this RNA exit channel. Clear? And therefore termination occurs. Clear? Termination occurs, and then this RNA polymerase will go out, or dismantle, and then DNA will again bound, and they will form again double helix. Clear? So this is row independent mechanism. Clear? So in row independent mechanism, what we see, that formation of hairpin loop is very, very important. Clear? So this is the basic thing you need to understand here is this. So these are alternative base pairing. This is what? Repetition of A-rich series or A-rich sites. So these two are very important for row independent mechanism. Okay. So we have successfully completed transcription in prokaryotes. Now we'll be talking about